it's worth a trip. So I'm going to indeed discuss today specificity of a T cell response and maybe challenging it a bit at the end of the talk. So the project, started when we decided that we wanted to analyze the TCR repertoire of Tregs. And the reason is that because Tregs are very important. As you know, they protect us from developing autoimmune disease. If you eliminate Tregs in an otherwise healthy mouse or a healthy individual, you will get immediately multi-organ autoimmune diseases and inflammation, and the mice actually will die when you do that. So they're quite important, these Tregs, to maintain a healthy immune system. And we thought that actually understanding what they recognize was one of the most important challenge in Treg biology. Since, of course, other questions like the mechanism of action, I think they're important, but we still don't know exactly what they recognize. So the project started trying to modestly define and analyze the repertoire of TCR recognizing insulin, for example, from mouse and human Tregs. And it all started first with in vitro methodologies to try amplify cells that are specific for these peptides, and then sequencing the TCRs, but we actually failed, and I don't have to tell you why here. So we then tried to find an alternative mean to explore Treg TCR repertoire, and we reasoned that maybe doing in vivo experiment would be more interesting and more efficient. And we turned to the setting of analyzing the TCR repertoire of follicular cells, T follicular helper cells, and T follicular regulatory cells. So what about these cells? Briefly, as you know, in lymph nodes, there are B cell follicles, and it's in the germinal centers of these follicles that actually the B cells that produce antibodies are actually generated, plasma cells, and where a somatic hypermutation goes on. And all start in the germinal centers with some T cells actually interacting with B cells and then triggering the proliferation of these cells and all the events that actually lead to antibody production. Going into more details, the interesting thing in this setting is that it's assumed that the Tfh cells, in fact, interact with the B cells being the antigen-presenting cells to these Tfhs, as we learned many years ago from Antonio, actually, that B cells are very efficient APCs. And this goes to triggering antibody production. So because of this interaction, we felt that the Tfh cells would be very specific for the antigen to which the B cell responds. And as we know, at least in theory, the antibody that are produced from this reaction are supposed to be very specific. So if the antibody are specific, the Tfh cells that recognize the same antigen should be specific. For the Tfr cells, it's a bit more complicated. We didn't know, but we assumed that it would be the same. Okay, so again, the conclusion from this was that we believe that looking at the TCR from Tfh and Tfr could be a heuristic for understanding the Treg TCR repertoire. So what did we observe from this experiment? Most of the results have been actually published recently, but they're the introduction for the second part of the talk, where I'm going to discuss how we interpret our results. So we basically first look at the repertoire of Tfh and Tfr using the usual means to first analyze globally the structure of the repertoire, the diversity. For example, here the reflection curves are clearly indicating that the repertoire of Tfh and Tfr is actually quite less diverse than the repertoire from Tf and Treg. And this is true both when you purify these cells. Actually, I should have said that we're using a spleen cell, not lymph node cell, but it's an approximation of the lymph node cells. And even if you actually purify these cells from a non-immunized mice, you would actually have this diversity profile of the rarefaction curve shown here. It doesn't change much after an immunization with either a self-antigen, which is insulin, or ovalbumin. You can look at other parameters of these TCR repertoires, like, for example, the P50, which represent the percent of the most predominant unique TCR beta that account for 50% of the sample. So the lower is this P50, the more expansion you're supposed to have in these repertoire. And again, here, these indexes are actually lower for Tfr and Tfh than they are for Tf and Tregs. We went on to look a bit in more details to this repertoire. For example, first, reconstructing from the TCR sequence data the famous immunoscope profile that we used to work with in the ancient times. And as you can see here, you immediately see that in Tfh, as in Tfr, you have a profile that shows some expansion. Here, for example, just 3 TRBV that are actually highlighted. And this is really in marked contrast to what you actually see looking at the Treg and the Tf profile. So there are some kind of expansion, and you can actually see this in a different way, as represented here. For example, when you look at the top 250 unique TCR beta, and you represent how much of the repertoire is their cumulative frequency, you can see that there are clearly expansions in Tfr and Tfh, actually about the same type of expansion compared to Treg and Tf, for which you hardly see an expansion. You also see that actually the profile that we're seeing here is very similar, whether you have no immunization or whether you have immunized the mice with INS or ovalbumin. You can also look at the global structure of your repertoire by analyzing all of the clonotypes and doing a PCA that will indicate that actually Tfh and Tfr have a global structure of the repertoire which is different from Treg and Tf, the Tf and Treg being different, with the impression here that the repertoire of Tfr and Tfh is very much alike. But if you redo the PCA, getting rid of the Treg and the Tf, you will see that they are indeed a bit apart, one from the other, which you could interpret as a kind of indication that there are distinct specificity in the immune response by Tfh and Tfr. But this is a bit of a loose interpretation. Now, to go deeper in the understanding of the structure of this repertoire, because it's of course important for what I'm going to show in the end, we tried to look at the sharing of these different repertoire one with the other. So what we're doing here, we are selecting the first 250 unique TCR beta from, for example, the first sample of TFRs, TFRs 1, and we're looking at how these 250 uTRs are represented in the other 5 TFR samples or in the other 5 TFH samples. And we have a cumulative frequency that we can then plot, which is going to kind of indicate the sharing or resemblance between the different samples. What you see when you do that is, first, if you just compare broadly, independently of immunization, the TFR and the TFH, you have the impression that the sharing is a bit more for the TFR with the TFR and the TFH with the TFH, but it's not spectacular. Then you can actually look and see after immunization with insulin or ovalbumin to see if you can get a hint about some sharing that is going to be linked to the immunizing antigen. So for example, in the mice that have been immunized with insulin, you look at the TFR cells and you look at actually the sharing between the TFR cells that have been immunized with insulin and those that have been immunized with ovalbumin, you can see that it seems that there is a bit more of a cumulative frequency here than here. So insulin with insulin, you can see more of the resemblance. And it's also the case for, if you look, for example, at TFH with ovalbumin, you have some kind of a higher sharing here, OVA with OVA than the rest. But you will admit that this is not exactly what we were expecting. We really thought that we would find a good specific response with actually a few clones and a lot of sharing. This is not what we're seeing. So because we didn't see it with this kind of analysis, we went to try force a bit the system. And for example, we look at the average cumulative frequencies of public sequences that are shared at least by 3 of the 6 samples we had per cell types and per immunizing condition. And when you do this, you see that actually the cumulative frequency of the public OVA uTRs from TFH is actually higher in the OVA sample from the TFH cells than in any other samples. And here you can see that the insulin, the public insulin uTR-B from immunization with insulin are a bit more expressed in these samples. So when you force the system, you have the impression that you can detect more of an kind of OVA-specific response in the OVA TFH and more of an insulin-specific response in the insulin TFR. So that was where we were when we decided to move to a different type of analysis. We thought that maybe we could actually use the notion that TCRs with the same specificity have very close, no, I should reverse the sentence, that TCR with very close sequences are more likely to have the same specificity. And we did what many do, which is plotting the structure of the repertoire based on the Levenshtein distance of 1, meaning that you allow 1 difference in amino acid between 2 TCR to actually link them. Okay, so 2 TCRs that are linked one to each other have just 1 amino acid difference in their sequence, whether it's in the middle or at the end, an addition or a deletion or a substitution. When you do this, and you do this, you can do it for the entire repertoire, or you can do it with the first 1,000 most represented CDR3s. Of course, you do it only for CDR3. You don't look at the rest of the TCR sequence. You see that for TF and Treg, you see that kind of representation, where you have a few independent clusters and then clones that are single, not connected. You can look here also at the representation of the abundance of each of these clones. And as you can see, being in a network in TF or Treg, whether you're from a control or in that case immunization with albumin, you still have quite a low frequency in terms of your representation. You only have a few, in fact, clones here that have a higher representation. Now, this is the structure that we're getting with Treg and Tf. And of course, what we were expecting looking at Tfh and Tfr would be, in fact, to have 1 or 2 huge clusters with cells that are for each of the nodes, making the clusters a much higher abundance, which is in fact not what we saw. What we saw is very few clusters, smaller, but even more important, we saw that there were tons of singlets that were not so, meaning so they were not connected to anything, and they have actually some kind of a significant expansion. So in the germinal centers of lymph nodes, where an ongoing immune response is supposed to lead to the production of very specific antibodies, if you look at the T cells that are there, and I forgot to say that we have markers that can, help us to purify the Tfh and the Tfr pretty well, meaning that we're sure that they are in the lymph nodes. In fact, for example, the expression of CXCR5, which is the attractor for these cells to put them in the lymph node. So in the lymph node, you see cells with unknown specificities that are not linked one to the other. So it doesn't seem to be a coordinated immune response, and that actually have some significant, although mild, expansions. So we were puzzled by this. We then wanted to go in more details into this interpretation, and we worked with Michael Shugai here and used TCRNET to study this a bit deeper. Without going into the details, here you have the frequencies of the TCR beta, and here you have the p-values that are computed for the degrees of connection. What you expect, basically what we expected to see if there were some kind of specific expansion would be clones around here. What you can see on these slides in the visual manner is that, for example, in TFH cells that comes from mice immunized with ovalbumin, you have actually some degree of connection that you don't find in the other settings, but not so many expansions. So you can't really see the marked specific response we are waiting for. And you can also see this on that slide, but for sake of time, I don't have the time to describe it. So the only thing here I just wanted to mention is this representation showed that in 3 different TFH samples, the response, in fact, that we detect is very private. Now, what are the provisional conclusions from this work? First, that a specific immune response to antigen is very difficult to detect in TFH and Tfr cells, to a surprise. The repertoire of Tfh and Tfr before as well as after immunization is poorly connected and mostly made of expanded, isolated TCR betas. And that to us, this suggests that there is a bystander activation in the germinal center, which is resulting in the moderate expansion of many stimulated clones that are not so specific for the immunizing antigen. So this is, then what was the starter for the thinking about what these results mean. And I'm going now to discuss how we interpret it, which is that there is a second immunologist's dirty little secret that has been ignored so far. So why a second? Because as you all know, there was a first dirty little secret that has been actually outlined by Charlie Genoway in a famous conference at the Cold Spring Harbor meeting. What he actually analyzed and showed and discussed is that you don't have an immune response if you just stimulate an animal with an antigen. You need actually to give something else, which is in fact adjuvant, to finally obtain that you have a detectable immune response, and which was, of course, the beginning of the development of the notion that you need the stimulation with the antigen, the TCR stimulation from the antigen, but also co-signals. At the time, he was mostly discussing IL-1, but we know that there are many co-signals. So now, what is the second immunologist's dirty little secret? Is that I think specific T cell response might not be so specific. Oops. And the reason, because we've ignored this for so long, is because, of course, immunologists have been looking at T cell response with tools that help them purify cells that they call specific, and these tools are actually tetramers. So let's first briefly discuss specificity. of course, we've all learned that there are two major properties of the immune response. One being that it is of the adaptive immune response. One being that it is specific, and the other one being that there is a memory. And in fact, memory is also linked to specificity because the memory is specific. And in fact, this paradigm is kind of validated by tons of knowledge. And for example, to me, I'm not questioning that there is specificity, because if you immunize, if you vaccinate someone against flu, that person will not be protected from measles and vice versa. So specificity does exist. But because of the importance of this property, the immunologists somehow have furiously focused on analyzing specific T cells, and whatever the methodology they use, cloning, tetramers, not single cell transcribing a TCR, they focus on this and they forget that there is something that you don't pick up and you don't analyze when you do so. So now, before I show you paper that data that actually question this notion of specificity, one also can wonder, how the immune system would have been selected during evolution to have a high degree of specificity. What we usually teach is that thanks to this fantastic, miraculous way of generating a huge diversity with the process of recombination that help generate the TCRs, you will generate such a diversity that, in fact, you will always have the proper specificity for a coming antigen. And even though there will be very few cells of a specific TCR, the process of their amplification will lead to an efficient response and also to the generation of enough memory cells so you have memory in the future. But this, in fact, is challenged by many recent studies, and I'm just going to show you, some are not so recent. I'm going to show you 3 pieces of evidence that actually challenge this, in addition to what I showed you earlier. First, this is a paper that was published now quite some years ago. I don't remember which year was that. 1999, yes. This is one case of a patient with an immunodeficiency, which in fact leads to the absence of, in theory, the absence of recombination and of thus B cells and T cells. And that person had actually one of his thymocyte precursors undergoing a reverse mutation that led to the generation of an entire repertoire from one single thymocyte, or the only one in which there was a reversion. And in fact, this is interesting because that person, so it's called an X-linked severe combined immunodeficiency, had this reversal, and it was estimated that there was, so at the time there was of course no NGS, that there were really a few cells with different TCR betas, but despite this very restricted repertoire, that person in fact did very well until, in fact, very well. It did not do so well because actually that person died after a few years, but it died from actually a B cell deficiency, not the T cell deficiency. And in fact, this patient didn't have any opportunistic infection of the kind that characterized T cell deficiencies. In fact, even had varicella with a normal outcome. And so it's clear that there was, with such a restricted TCR repertoire, there was a good enough efficacy to mount immune response against different pathogens. So the idea that we need to generate a huge diversity to efficiently fight pathogen is somehow a question here. The second work I'd like to show here, and I'm close to finish, is a paper published in 2015 where the authors, studied different peptides recognized by the same TCRs, and then the functional relevance of that. They showed that, in fact, in their case, that having in common 5 amino acids, but not contiguous as it was proposed earlier, was sufficient to bind the same TCR. And they showed that this is functionally relevant because with different peptides that recognize the same TCR, they could either create autoimmunity or actually mount efficient immune response against foreign peptide. And I'm just going to illustrate this with one slide from their work. So here, the self-antigen is MOG. It's from neural antigens. And if you look with a tetramer that recognize the MOG antigen present in the right with the right class 2 antigen, you can see that before immunization, you have very few cells. You immunize with MOG, and you have actually a very nice response with a tetramer. And usually scientists do what they do, they just purify this and they analyze this. But the thing is, if you immunize with actually 3 different peptides that come from different pathogens this time, so this is a self-antigen, these are peptides coming from pathogens. Their sequence is represented here. They always share 5 amino acids, as I said earlier, but have a marked difference in the rest of their sequence. They actually induce almost as efficiently these so-called MOG-specific cells than MOG itself. And now, if you use these peptides to try induce a pathology, which is called EAE, which is supposed to model MS, with MOG peptide, this is the course of the disease. This is the score for the clinical score. But you can also actually generate a significant pathology with these peptides coming from really different sources. And finally, just to close on, there was a paper which I think maybe was not so well noticed some time ago, also in 2015, where Mark Davis and his team reported that cytomegalovirus infection enhances the immune response to influenza. Okay, what they showed in this paper is that when you've been exposed to CMV, you have all kind of modification in your immune response parameters, where they have a proportion of different cells, etc., etc. We've not looked at the TCR, but this will prepare you better to respond to influenza. They also show that experimentally in mice, you can also show that pre-immunization with one enhance the response to the other. And in the end, they interpret this as the fact that CMV infection somehow you respond to it and it prepares your immune system to better fight flu, but they don't speak at all about specificity. Now, how do I interpret all this and my own understanding of things? First, and going back to the experiment looking at Tfh, Tfr, this major bystander activation of Tfh and Tfr that may not be specific in the usual sense to the immunizing antigen, maybe is due to the fact that when you think about it, the activation of the T cells is one through a so-called specific signal that goes with the TCR, but second through the integration of multiple signals coming from all kind of other molecules, whether they are cytokines or other molecule expressed by APC. And all of these second signals are non-specific. So when the T cells enters an environment where you have all kind of cytokines present and all kind of cells that are more or less activated, express these molecules that can actually provide second signals, the signal from the TCR is only adding to these other signals that are non-specific. And in the end, to be activated, maybe you just have to go through an integrated threshold that is not, doesn't need to be so specific. And I'd like to propose that, in fact, what's going on is the fact that you only need a kind of a fuzzy specificity to be activated, meaning if your TCR recognize something but the signals that are around are strong enough, you're going to be activated. I think that this notion that you don't have to select for cells that are so strictly specific for an antigen has some meaning that we can then discuss for the selection of a functional immune system during evolution, because, you know, and this is something we can discuss after, I think there are no good explanations so far for why you're both being, what has been selected is both this diversity and the MHC, which is something we can discuss. I think that when we look at convergence in the immune response, as it was discussed this morning, we should not just look for shared TCRs, but something a bit broader. I think that, of course, this fuzzy specificity could be somehow linked to the development of autoimmune disease. And I showed you some work in the Nelson paper that clearly indicate this. You also know, for example, that in type 1 diabetes, there is quite clearly, or at least an infection event that triggers the disease is anticipated and looked for. And finally, I'd like to remind you that there are not just T cells in the immune response. And in fact, the specificity we like to discuss is maybe mostly carried on by antibodies. And also it's something that I've not heard discussed often. Why is it that the T cells have not retained the somatic hypermutation that characterize B cells? If there was really a need for a very specific response through the T cell receptor, this would have been actually a great system to use to. And with this, I'd like to leave you with this hypothesis, and thanks, the collaborators in my lab and Michael Shugai who collaborated with us. Thank you very much.